Hello everyone, welcome back to another BGCSE past paper review. Today we're reviewing a biology paper. This is a paper 5, otherwise called the alternative to practical. This is for students who do not have access to labs or taking the exam privately or decide to not to do the coursework based upon various reasons. Again, before I start this examination, please write your school number, your candidate number, your surname, and your initial. This is 2017 paper, by the way, just to make a note of that. Again, this will be done just after paper 3. So it's be on the same day as paper 3 usually. And it's a 4-question paper. So please, you need to read through the instructions carefully and make sure you answer all the questions that it explain or state that you need to answer. All right, and so let's jump into the first question now. And the first question says diagrams below show the materials used in the experiment or in an experiment to compare the energy content of a peanut with a cashew nut. All right, and so here now we have our beaker of water, we have our tripod stand, we have our nut on a mounted needle, and we have our thermometer. Now in the space below, draw a diagram to show how the apparatus would be set up to measure the energy release by the nut when burned. All right, so now I have a diagram there already. I put them together for you to save some time. So you see the diagram is right there. So the nut, if you notice, the nut will be underneath the tripod, the beaker on the tripod, and the thermometer in the beaker with the water. All right, so let's go into answering the question now. It's a give a matter of fact, if you can look under the biology playlist, you'll actually see an experiment just like this I've done, but I've used a different method. I use a pin, hold it slightly underneath the test tube with the thing. So there are different ways you can do the same experiment, right? All right, so part B here now said give the precautions to ensure that the thermometer reading of the temperature of a liquid is accurate. And so there are a number of things that you need to do right here. I'm going to give you more than 2. So one of the major things to do is to read the thermometer at eye level. So read at eye level. I'm doing this I'm just going short as possible. Read at eye level, all right, to avoid parallax, okay? To avoid parallax. All right, so just to make sure that you understand. Parallax is an error. So let's say parallax error. That means like you're reading, you're gonna see like if your eyes aren't at eye level, you may have a point below or a point above, depending on how you slant your eyes. So your eyes must be at eye level for one. All right, the next thing here that you can have is that you should not, the thermometer should not touch the bottom or the sides of the beaker, okay, while taking temperature. While taking the temperature. All right, because if the thermometer is touching the sides or the bottom of the flask, then you're actually reading the thermal conductivity of the flask rather than the liquid itself. All right, so the temperature of the flask may be different from the liquid in the flask itself or the beaker. So be careful with that. All right, so the next thing here now is that you're going to keep the thermometer. Let me just write this here. You need to keep, so let's call this number 3. Number 3, keep the thermometer straight. Keep it straight. All right, keep it straight or upright. Keep it straight or upright. All right, let's say straight upwards, right? Let's say straight upwards, upright. I keep it straight or upright, and that is to ensure that the expansion of the mercury, if it's a mercury thermometer, so the mercury could expand equally or evenly and not slanted. So the slanted going to be rushing faster. If it's straight, it takes the equivalent required amount of incremental increase. If it's slant, they know, of course, if liquid is slant, it run fast. If it's slant and if it's straight, as we know that, right? And next thing again that you can do as well, this is a 4th one, is that you need to clean the bulb. So you clean the bulb of the thermometer. And the reason why you want to clean the bulb of a thermometer, because if a bulb has residue, then it's going to affect how the thermometer conduct heat or heat transfer to the thermometer itself, and it may not show accurate reading. So clean the bulb of the thermometer. Okay, you need to do that. For best, let me say for best conductivity reading, for best conductivity slash reading. All right, because remember, heat must be transferred to the thermometer for you to read it, for you to read the temperature changes, right? And if it's not done, then you're going to be in some problem. All right, so part C now, we say a cashew nut contains more fat than a peanut. Predict how the result of this investigation would differ for the two types of nuts. So if cashew has more fat, remember now, fat contains more energy per gram, right? So if cashew have more fat now, what's going to simply happen here now is that the cashew will burn longer, the cashew will burn longer, all right, longer, and give a higher temperature reading. All right, so yeah, so the cashew will burn longer and give a higher temperature reading for sure because it contains more energy. Okay. All right, for part D now, is to give one reason why why is it important in this experiment to use the same volume of water for each not tested. And we can say it in many ways, right? Because again, if you have a small amount of water or different volumes of water, the smaller volume will heat faster than a larger volume, right? So you need to make sure consistent in heating water. So to ensure consistency in eating, that's pretty much it. All right. So to maintain consistency in heating. All right. Or a next way to say this, I must put it like that. A next way to say this is to ensure that the heat change is related to the nut and not the water. All right. And so let me say to ensure that the temperature change is related to the energy in the nut and not the water. Okay. All right, because different volume of water give you different heating time. Larger volume of water take a longer time to heat up, right? And we know that if you can cook, have a small pot of water, boils fast. If a big pot of water, take a longer time to boil, right? Yeah, so you want to maintain consistency in heating. All right, so that's pretty much it for that. All right, it said the same mass of cashew and peanut. And so if you say to have the same mass of cashew and peanut, this is actually compare energy per mass, okay? All right, and this again is also for consistency purpose. To compare energy per mass, okay, to compare energy per mass, or simply to obtain a better understanding of the amount of energy per molecule. All right, to understand or to obtain, you may say how to say that. So it depends how you see. You can say it in so many ways. To understand or to better understand. Let me say to better understand. All right, to better understand or to obtain accurate data, to better understand or obtain accurate data about the amount of energy, per molecule. Okay, because remember that mass is the amount of matter in the thing, right? So per molecule of nut. Because bigger nut, if you have a bigger nut, it's not going to tell you anything. And matter of fact, for nutritional values, for example, you want to know, okay, if I eat the same amount of nut, which one will I get the most energy out of? Rather than eating a big, like for example, a pecan versus a peanut. Obviously, one is larger, but it doesn't mean that it is more nutritious. And again, you may eat a small amount of nut, get the same amount of energy. Right? So things like that are very important. So we want to make sure we understand the amount of energy per molecules. So if I have to have a discussion or make a prediction, then you can say, okay, if I have 10 grams of peanut, then this would be the energy. Whereas if I have 10 grams of pecan, this would be the amount of energy. So you have better predictions for the results as well. So you can make better prediction, or you can analyze the data better. So I can make even a note right here for a better analysis of the data. All right, so I'm gonna make a note right here. So there's so many things you can write here for better analysis of the data. Okay, yeah. So there's so many things you can write. My next thing right here is to place each burning nut at the same distance from the water, and this is to ensure consistency again. Consistency. So it's about consistency to do all these things. And these are what, if you do an experiment, they call these the control variables, right? So to ensure consistency in the rate of heat transfer. I want to say rate of heat transfer, I'm not saying about fast, but in terms of distance. If one is further away from the beaker, obviously the heat going to take a longer time to reach it because the distance is so far, right? So you want to make sure that the rate, that means you have the same distance and the flame is burning, one is not far, so the radiation. So let's say heat transfer radiation, right? So heat transfer, let's put radiation in bracket. So you want to make sure that the radiation is the same or the heat transfer is the same. Okay? So radiation. All right. And so to ensure consistency in the rate of heat transfer, so that is also important there. All right. All right. So let's look at the next part now. It said, why would it be useful to repeat the experiment? And this is nice. This is a nice thing to do. You want to increase accuracy, all right? To increase the degree of accuracy. That's simply it. To increase the degree of accuracy. Yep. So you do it more than once, take an average is a better prediction. Not only that, what this also does is to decrease. Let me say here, to decrease anomalous results. Anomalous. All right. So to decrease anomalous, I think I have an A there. Yeah, this need to come out to be an O. Right. Anomalous results. And anomalous result is really to exclude or to reduce those errors or things that may affect getting accurate results. So the more you do it, if there's any change in the environment, that will reduce the impacts. At least doing it more than once on different conditions or the same condition, you get a better accuracy in terms of a greater prediction, a better estimate of the true thing. All right, so that question is out of the way. That's the first question done. Question number 2 now. It's a diagram shows 3 tests used to identify presence of specific nutrients in food item. Now, here now it said iodine. You have iodine. Iodine is testing in a thing right there. That's for test 1. So test 1, let me just highlight as we go along. Test 1 is iodine test. Test 2 is Bendix reagent. We know that is test 4. We have copper. This is the same thing. Test 2 is this. Let me just put an arrow right there. So you do this with the Bendix, then you do this. Then test 3 now, using copper sulfate and sodium hydroxide. All right, most of you may not actually know what this is. What this is related to is equivalent. It's not the same as Biret, but it's similar to Biret because Biret will have some other stuff into it, right? Let me not get into that too much, but Biret will have some stuff into it. It's similar to Biret in terms of reaction. All right, so it's equivalent to Biret solution. Let me say equivalent in terms of test. Okay. Biret has more stuff in it. All right, just to point it out. So here, this one is the equivalent to. Let me put a star. And so you should know that this is equivalent to Biret. All right. So Biret solution. It is equivalent to Biret solution. All right. Solution. All right. Just to make a note right there, equivalent to the Biret solution. All right, so let's see what the next part of the question asking us now, and we should be good. All right. So here now, in the next part of the question, it says we have the name of, it says we have a test 1, 2, 3. We have the name of reagent, initial reagent color for positive result, and then we have here now nutrient identified. So for test 1. If you have iodine brown, it was brown before, then if it's a positive test, we're going to get blue, black, or black. So just say blue, black, or black, don't matter. All right. All right, so get blue, black, or black. Nutrient test for your starch. We know that. All right, so this was an easy test right here. For test 2 now, we was using Bendix. So I could just put Benedict's right there. So it is Benedict's solution. All right, the Bendix solution. Let me just put that SOL right there. And the color of Bendix solution is blue, and the change to orange test for reducing sugar. Copper sulfate and sodium hydroxide is also blue, just like Biret. And the positive change here going to be purple. So you should get purple if you get protein, and protein is what you test for. All right, so it's equivalent to, so if you didn't know, it is equivalent to Biret solution. Oh, matter of fact, they have it in bracket here. Excellent job. Excellent. I never realized that, but awesome. All right, so it's equivalent to Biret. Right, cool. But Biret, industrial Biret or laboratory Biret do contain some of that stuff, right? What is equivalent? All right, so part B. It says state one safety precaution carried out when performing test 2. And test 2, I don't need to go back to the diagram. The table will tell me. Yeah, test 2 is using Bendix solution. Bendix solution use some heat, right? So let me just quickly tell you this, that during eating, so while you're eating this, while you're eating up the stuff, let me show you what it is. So you see exactly what I'm talking about. See, test 2, apply heat. Okay. All right. So just to make a note right here. So during heating, turn the mouth of the test tube, away from self and others. And if you do this in my lab, turn the test tube to yourself and others, I'm giving you 0. All right. So thank God you're not in my lab. If you do foolishness, I'll be mad. I'll be so mad because it's dangerous. All right. And I don't want to play in labs. You should not play in labs. You all should know that. All right. So next thing you know, move the test tube slowly through the flame. Okay, slowly through the flame. And so let me just make a note here. Do not hold in one place. All right, so do not hold it in one place at all. You move it slowly through the flame. All right, and the purpose of doing this, let's hold the red place and put position. I think I prefer position. Do not hold it in one position. All right, and yeah, and the reason for this, just to make a note, the reason for this is to ensure, in moving through the flame, to ensure equal heating of the test tube. All right? Yeah, so equal distribution of heat across the test tube. That's very important. Otherwise, the test tube can break. Matter of fact, not only that, you can have violent reaction, or violent boiling, I should say, and then liquid could spill all over the lab, and then liquid spill over the lab, I get mad, and then we have problems and things like that. Anyway, you can damage yourself. That's the most important thing. Don't mind what I'm saying. Yeah, but you can cause damage to yourself and your lab materials, and you can contaminate your solutions, you contaminate your chemicals or whatever you're using. You can mess up a book, mess up a uniform, even though you should have lab coat. Liquid could spill the liquid could spill in your eyes, even though you should have goggles. All right, so things like that. You should have a safety gears. But sometimes, I'm not saying anything. All right, part C. Part C1. Sodium hydroxide is a corrosive chemical. What does the term corrosive means? Now, corrosive means now that it causes damage. Causes damage by chemical reaction. When in contact. All right, when in contact. All right, good. That's simple. So it causes damage by chemical reaction when in contact. And some of the example here would be to the skin. So example here to the skin, and even metal or metals, right? And if you look at a corrosive sign, you actually see the metal sign there that it burn out piece of a metal and also burn out piece of the skin. Yeah, so corrosive, it could damage your skin and metals. All right, so here's to give 2 safety precautions. Precautions used when handling corrosive materials, and one is to wear your goggles. So wear safety goggles. Don't play in the lab. I'm not going to write that, but yeah, don't play in the lab. You should not play in the lab. Don't play around in the lab at all. Wear safety gloves, and lab coat. All right, so don't play in lab for sure, don't play in lab for real, because playing lab could be dangerous. You could have spillage, you could slip, you could burst your head on the counter. There's so many things, you can die. So don't play in the lab, don't play around. The lab is a serious place. Do not play in the lab because you can spill chemicals over to another children. You damage yourself and all that. All right, so question number 3. Third question. Third and second to last question. An experiment was carried out to see the effect of 3 different solution on potato strips. So we have a potato right here, first diagram potato, and you have a rubber borer or a cork borer to punch out the strips of the potato. These are the strips of potato here. Maybe not so brightly seen, but anyhow, not a problem. You have water in one beaker, you have 1% sodium chloride, which is salt, 2.5% sodium chloride here, which is salt again. All right, so concentration increase as moving from left to right. I see that is important there. Is that all strips were cut to 40 millimeters long and placed one in each solution on the left for 3 hours. They were removed and on blot dry. The strips are shown below. So strip A, strip B, and C. And here it said, measure the length of each strip and record your result in the table below. All right, so now I've gone and measured all of them already. So I'm going to tell that strip A is 40. Strip B is 22. And C is 26. Okay, so I've valid measure those. All right, no need to waste your time watching me doing it. Is to calculate the percentage of each strip shown, show you working. So to calculate the percentage change, look, we say percentage change. So watch me now carefully and follow me. All right, the percentage is the difference over the original, right? So look at the first one, 40 original, 40. So the difference over the original times 100%. So for this one now, we're gonna say, okay, 40 minus 40, and it's a show working now. So not to write down the number and those things you're gonna get marks. No, show you working. Even though you know it's 0%, and I know you're smart and you know that, it's a show working. And notice this is 6 marks. So if you don't put 0, you get 1 mark. All right, you need to show you working. So that's equal to 0. Difference here is 0. And so we're gonna say 0 divided by the original, which is 40, right? Multiply by 100%. So we're going to multiply this by 100%, and that's going to equal to 0%. See, 0%. That's easy. 2 marks right there. Next one for next 2 marks. You're going to say 40 from 22. All right, so go back to your chart. I remember them, so those no need to stress. Just 22 and 26, all right? And all of them start at 40. Okay, so we're straight. So next one now, strip B now is 22. Is let's say 40 minus 22. All right. 40 minus 22, and that gives us 18. Yeah, that's 18. Correct. Yeah, 18. So we're going to say 18 now, which is the difference there, divided by the original, which is 40, multiply by 100%. So that's doing this 3 times to get 6 easy marks. So lucky. Equal to 45%. All right, we'll go to the next again for next 2 marks. Oh, geez. All right, so this one is 46, right? Sorry, not 46, 26. So 40 minus 26, and that's going to equal to 14. And so therefore, again, it's going to be 14 over the original, divided by the original, which is 40. All right, and then multiply that now by 100%, and that equals to 35. That's 35%. Yeah. All right, so we're straight. So that's 6 marks. Easy mathematical marks. All right, so explain your answer for strip A. Now, look at strip A carefully. Strip A, increase, yeah? Okay. No lie. Strip A stays the same. All right, so strip A stays the same. So explain the answer for strip A. Why strip A remain the same? Simple start out by saying it was placed in an isotonic, right? Isotonic solution. All right, so place an isotonic solution. But it simply means that the concentration in the potato is equal to concentration in the beaker. So the concentration in the potato equals the concentration in the beaker. Okay? Yeah. All right. And let me say something here. Therefore, there's only one mark, but I'm just giving you some things you can say. Therefore, there is no net movement, movement of water in or out, of the strip. So, okay, so water leaving is equal to water going in, all right? Because it's isotonic solution. The concentration are the same. So there's no movement of solution, of water, I should say, movement of water. So concentration are equal, water entering equal to water leaving. And that's another question that is done. This paper is nice and easy. Question number 4, final question. It says diagram shows the steps in an investigation set up by a student in a biology class. All right, so it said here now, okay, we have a 1, 2, 3, 4 things. Okay, what does it say now? It's a name process being investigated here, and the process that is being investigated is actually transpiration. But I want to tell that this thing don't really measure transpiration exactly in it, actually measure the rate of water uptake, right? But the process is transpiration, because transpiration is what causing it. So I'm gonna put in bracket here just to know the title. If you're doing this experiment at school, or if they ask you to explain what is happening here, is that actually measuring the rate of water uptake. Okay, so measuring the rate of water uptake by the plant. Okay, because remember now that when water uptake by the plant, the reason I'm telling this is all photometer, matter of fact, does not accurately measure the rate of transpiration. What they actually measure is the rate of water uptake. Because truthfully, even though this is measuring mass, when the water goes in the plant, some of it will transpire for real. You have transpiration taking place for truth, but some of the water will also use for photosynthesis. And you may say, okay, but if the use of photosynthesis is the molecules are still in the plant. But remember, during photosynthesis, oxygen is given off, which is also a loss of mass as well, yes? Because oxygen do has a mass, isn't it true? Yeah. So it really measures accurately rate of water uptake rather than the rate of transpiration. But the process that is really measuring is transpiration. Because transpiration also makes water available for the plant, okay? For photosynthesis, isn't it? All right, so just to mention that. So if you have to explain this like a real good 5-mark question, I want to get into it and you start talk about why it measure the rate of water uptake rather than the rate of transpiration. You need to know that type of stuff, you know? You have to really dig into these things. All right, so using the information shown in the first step on the diagram, write a simple method to show what it is, to show what is being done at each step. All right, I don't need to go back and forth the diagram, but just watch carefully. For your purpose, take a mental picture. Number 1, number 2, number 1, number 2, number 3, number 4. All right, so take a mental picture of that. Or you can go back and look at it and then pause and observe it and then come back to make sure you understand what I'm going to do now. So the first thing, number 1, let me put 1 there. The first thing in number 1, and this is a 5-mark thing, yeah? So you're going to see a plant was placed in a beaker with water, right? In a beaker with water. All right, so that's the first thing, a beaker with water. First thing, number 1. Number 2, 5 marks. Just look at the picture, write down steps. Lucky. All right, oil was poured. All right, so oil was poured in the beaker to cover the surface. Only the surface we're going to cover now. Or to cover the surface of the water. We don't need to pour it and fill up the beaker with oil. Just to cover the surface of the water, right? 3 now. Third marks. You're looking for 5 marks. 3 marks. The third mark, or the third point, is that the mass now, the mass of the beaker, the mass of the beaker, let's say beaker with water and oil, yeah. Must make sense. The mass of a beaker with the plant, water, and oil was measured. All right, so measure the mass of the beaker with all the things in it, with the plant, the water, and oil. That was measured there. The next thing now is a 4th mark. Is that now a fan? You notice the picture there in the 4th diagram. A fan was used. All right, was used to blow wind across the plant, across the plant for 30 minutes. All right. Number 5 now, fifth mark. All right. The mass, or let's say the new mass, because this is a new mass, all right? Okay, quote unquote new, because, or the final mass, you can say the final mass, the new mass, whatever, right? The final mass or the new mass of the apparatus, let me say of the apparatus, because we know what in the apparatus already, of the apparatus was taken after 30 minutes. All right, and that's it. That's the steps right there. 5 marks. All right. All right. Next now, part C of the final question. It says, state the purpose of the oil in the investigation. And the purpose of the oil is to prevent the evaporation of water from the beaker. To prevent the evaporation of water from the beaker. So why this is important, right? So you only want water to evaporate from the beaker, right? So that's something I'm going to look at notation right here. So what is only being lost from the plant? So that's a little notation there. So any water loss is due to the plant, not the evaporation from the flask itself, only from the plant. So any water loss. So if you measure any water loss at all, is because of the plant is transpiring, not because of the beaker. And not covered with oil, water evaporate from the beaker. You can't say it's actual transpiration because you really don't know. So water only exit the apparatus through the plants only. All right. So here we said what happens to the mass of the plant over 30 minutes decreased. It decreased. Simple that. Just that. 1 mark. Yeah, the mass get less. If you look back on the diagram, you're going to see it. Explain why this happened. And it's simple because water evaporates from the plant. Okay. So water evaporate. Let's put transpire right here. All right. Transpire from the plant over time. Okay. All right. Over time, let me say over time, decreasing the mass of the apparatus. Yeah. Easy mark. That's a 1 mark too. All right. So what evaporate from the plant is good enough. All right. It's a calculate the change in mass during the investigation. I just want to show you something quick, fast and easy right there. See, this is 1423 and this is 1400, right? 1400, 1423. All right, so the difference here is, calculate the difference here. It's going to be 1423 minus 1400. That equal to 23 grams. I'm not saying grams. I assume that this scale is in grams, right? Then it's grams. So you're not going to be no kilograms. It's not going to be too heavy. Oh, that's his finish. End of the paper 5. All right, so thank you for being here. Good luck on the examination. See you soon, all right?